So, okay. Hi, I'm Ethan Viles.
And I'm Gwendolyn Cooper.
For the last year, we've been working on a study under Alyssa K. Hahn. We've worked on a pilot study studying the effect of microgravity on chondrocyte metabolism.
Do I go now? Travel is necessary for understanding the universe, physics, as well as analyzing if we can potentially one day do interplanetary travel. However, there are a lot of potential health risks associated with space sojourns. The two biggest ones that we have identified have been microgravity and solar radiation, and this study is focused heavily on microgravity, specifically the musculoskeletal system, and this is because these tissues are very sensitive to mechanical changes. So previous studies have looked at how chondrocytes, the cells found in your articular cartilage, have upregulated genes involved in the cytoskeleton and reorganized their cytoskeleton. And this is really interesting with just short exposures to microgravity. So what is really, really cool here is there's a lot of overlap between those results and results seen with the degenerative joint disease osteoarthritis. So this overlap means that there is potential in creating an in vitro model for analyzing both microgravity and osteoarthritis for the future. So the big goal of this project was to provide and complete a protocol that enabled us to encapsulate chondrocytes in a 3D gel agarose construct, as well as analyze their metabolic shifts. In our experiment, we had to culture our cells. So we used the human chondrosarcoma cell line SW1353, and we cultured these cells in DEMA medium, and they were maintained at 70% confluency throughout the study. Next, we had to make our agarose gel constructs. So we ended up counting our chondrocytes, making sure we had enough of them in the first place to go on with this study, and we used a 4.5% weight by volume agarose with a density of 1 million cells per milliliter. Next, we had to set up our rotating cell culture system. This involved inserting the medium into the vessels, making sure that they were all snug, and then turning it on and making sure that the speed was correct so that all of the cells were in a microgravity environment. These had to be maintained daily and observed daily to ensure there were no bubbles and that they were constantly in this microgravity environment. Next, we did confocal imaging. We took our samples out and we drove them to MSU, Montana State University in Bozeman, and we performed this imaging, which was really cool. And then we performed our data analysis using a metabolomics program called MetaboAnalyst.
Now let's jump into our results. If you look at the bottom box of figure 1, there's confocal imaging and data analysis. So that's what I'm going to be covering right now. Let's start with confocal imaging. Confocal imaging was used to confirm the presence of live cells after our experiment to ensure cells were still alive after the 4-day exposure to microgravity. If you look at figure 3, there is a red cell and a green cell. The red cell is dead and the green cell is alive. This confirms that there was alive cells throughout the duration of the microgravity experiment and after. And sadly, there was not a significant enough amount of cells to perform a complex statistical analysis on it. So right now we're just using confocal imaging to show that our encapsulation protocol worked and that we had alive cells throughout the experiment to analyze the metabolic or metabolite extractions. Next, for our metabolite extraction and data analysis, we essentially have 4 different cohorts of metabolite extractions. We extracted metabolites from the gels themselves and the metabolite excretions from the gels, that basically the media. So we have experimental gels, experimental metabolite extractions, and control metabolite gel extractions, and media control metabolite extractions. We can see in figure 2, we just compared every single gel versus all the media metabolite excretions. So we can see here is that gels were pretty distinguishably separable from the media metabolite extractions. In figure 5 and figure 6, we also did PCA, and we can see in figure 5, which is experimental gel metabolites versus control gel metabolites, that they're not easily separated using PCA, And we can see the same thing in figure 6, which goes over experimental metabolite excretions, so the media, and control metabolite excretions. Figure 4 actually shows a volcano plot analysis of all the metabolites found between all gels and versus all media. So this includes, in the all gels, this includes all of the control experimental gels versus all the control and experimental media or metabolite extractions. We see here in the graph, these pink dots are all significant, and then everything on the right side of the graph would be upregulated and everything on the left side of the graph would be downregulated. So these are metabolites that could be found upregulated in the gels and downregulated in the media. Goals of this study were to first develop a new protocol for in vitro microgravity exposure of chondrocytes, and the second goal was to study the metabolism and the metabolic response and changes to microgravity exposure. So regarding the first part is the most significant part of our study is that our procedure was a success. We were able to encapsulate chondrocytes in agarose, create agarose gel constructs, and then introduce these to microgravity environment for 4 days. And after those 4 days, we would have alive cells, and we could extract metabolites from those cells and extract metabolite excretions from the media and analyze those using metabolomics. For our second part, looking at the metabolic response and analyzing all the metabolic data that we got, it was successful. We were able to get metabolites, we were able to extract metabolites from the constructs. However, there are some limitations to that we'll go over later, specifically sample size, and we would just like to see more samples to confirm the data that we found. What we found so far suggests that microgravity exposure of 4 days, so short-term microgravity exposure, does not significantly change chondrocyte metabolism.
Although overall our study was very successful, we did run into some potential limitations. The first of that being was that we had a very small sample size, and this was due to having to refine a lot of our protocols to make them as efficient as well as effective, and so there was a lot of time spent there. Another issue we ran into, which we can see with our confocal imaging results, is that we just didn't have a high enough density of cells, and so that result ended up being much more qualitative than quantitative, which was kind of a bummer. However, it did still tell us that we were successful at encapsulation. It could also help account for maybe why our metabolic shifts or responses weren't exactly what we were expecting or what we had hypothesized. And lastly, and the last issue we really ran into was that our rotating cell culture system has 4 vessels that the construct and the media go into respectively, and they don't have their own independent motors, so this made it really difficult to ensure that they were all in a microgravity environment at the same time with the same speed. And so for future studies, we would recommend these alterations. And that is our presentation. Thank you for watching on this virtual surf. We'd like to first acknowledge Dr. Alyssa Hahn for being a fantastic mentor through this entire process. And we'd also like to thank Montana Space Grant Consortium for the funding for this project.